Hello everyone, this is Dr. Vishal Trivedi from the Department of Biosciences and Bioengineering, IIT Guwahati, and welcome to the live session for the course Molecular Biology. And if you have any questions, you can address. You can post your question in the chat box so that we will have a fruitful discussion. Good afternoon, Ronak. Can you post your question or any query or anything which you want to discuss with me? Good afternoon, Prem Kumar. Good afternoon, Aishwarya. You have any question? Please post your question. Good afternoon, Gayatri. Shilesh, Shilesha, right? Can you unmute and introduce yourself? Okay. यहाँ नहीं आ रहा इंटरनेट,
एक्सेस नहीं है।
अनआइडेंटिफाइड नेटवर्क बता रहा भाई। ये अभी नहीं,
ये तो नहीं दिख रही है इधर।
नहीं यहाँ नहीं आ रहा ना देखो।
ये सर ऐसे दिखाता है लेकिन लैन में कनेक्टेड है ना सर। अरे तो ठीक है सर, इसका नहीं है।
So all the participant, I think we have a technical issue at our end. So we'll sort out this issue and then we can be able to hear you. So just wait for a few minutes. Are you getting my voice, participants?
हेलो चेक, हेलो 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 चेक, हेलो। ये तो सब ये ऑडियो तो है।
तो माइक्रोफोन का है ना?
हाँ सर, माइक्रोफोन ही है। इससे ठीक है सर, अभी कॉल नहीं है। बिल्कुल, मैं मीटिंग में ज्वाइन कर लेता हूँ अपने से देखते हैं सर। ठीक
खोलिए सर। मीटिंग तो मुझे तो मीटिंग आज प्रियंका
बहुत दूर है यार ये वाला। तो रेडियो जो बताया ना यार, कैमरा ऑफ कर देता हूँ, फालतू में मेरा रिकॉर्डिंग।
Hello. कलर आ गया.
Hello, sir. Can you hear me?
Hello, Kalan?
Yes, yes, yes.
Kalan को बोलो बोलने के लिए.
हाँ सर.
Okay.
Hello. Vishal sir, I can hear you.
Okay. So, I think I will take the first. I think I will interact with the students which are on the team. So, can you unmute yourself, turn on your camera, and introduce yourself? So, we'll start with Suchi. Suchi, can you turn on your camera and unmute and introduce yourself? Arshita? Okay, I think let's take few questions from here, right, from the students. So I think the first question is from the Ronak. He said that what is the role of ribosome tRNA and proteinaceous factor in the process of protein biosynthesis? So Ronak, I think we discussed in detail about the role of these proteinaceous factors, role of these factors. So I think if you see, so I'm just sharing my screen so that it would be easy for you to understand. And okay, so I'm sure you can be able to see my this screen, right? And so we have three things, right? Ribosome. So ribosome, the function of the ribosome is that it is actually going to read, or it is actually going to read the tRNA on one side, so that it can actually be able to, and on the other side, it is actually going to read the messenger RNA. Okay, so it is actually a kind of linking between the two species. So you know that the messenger RNA is going to carry the nucleotide sequences, or I will say a message, right, which actually need to be carried, and then only the machinery will know what sequence the amino acid has to be attached, right? And then the tRNA is actually a carrier, so it is actually going to carry the uh, amino acids, right? And the carrying of the messenger amino acids would be in the same order as the message what is present on the messenger RNA. And the ribosome's job is that it is actually going to read this message, and according to this message, it is actually going to allow the docking of the particular tRNA. And then it is actually going to have the machinery so that you can actually be able to make the peptide bond between the amino acids. Okay. And as a result, what will happen is that amino acids are actually going to be attached to each other, and that's how it is actually going to form the proteins. Uh, Within the ribosomes, it will have the lot of proteinaceous factors and also the ribosomal RNA. So if you see the ribosome is made up of the ribosomal RNA, and it is also going to be made up of the different types of proteins. And apart from these proteins which are important for the ribosome synthesis, there are proteins which are actually going to be participate into the protein synthesis. So overall, if I summarize this, ribosome job is to read the message onto the messenger RNA, interpret that messenger that information, and then it is actually going to bring the the particular tRNA in the same sequence. And once the tRNA comes, then tRNA is actually going to carry the messenger RNA on going to carry the amino acids, and its another job is to form the peptide bond between the amino acids, and ultimately it is actually going to be lead to the synthesis of the proteins. Now, Ronak, is it clear to you? Okay, so it's clear. So now I'll take the another question, and another question is from the Nandana, right? So I think she is asking about the difference between the gram-positive and gram-negative bacteria. So I think the gram-positive and gram-negative bacteria, there are multiple differences. So I've changed the color also because some people are saying that the color is not very visible. So one of the major difference between the gram-positive and gram-negative is the cell wall, right? So cell wall is different between the gram-positive and gram-negative bacteria. And because the cell wall is different, it is actually going to show you the differential behavior towards the gram staining. And apart from that, the gram positive and gram negative, because the cell wall is different, it is difficult for the gram positive versus gram negative. You are supposed to devise the different types of strategies for the cell lysis, and you also going to devise the strategies for treatment, because if the cell wall is thick, then they are actually going to be more resistance for the antibiotic treatments and all those kind of things. So these are the some of the changes. We cannot discuss each and every differences what we have discussed in the course itself, but these are, mainly the cell wall is what the difference is between the gram positive and gram negative bacteria. Then we have another question, and that question is from okay, so there is another question from the Sormita Das, that which one is more popular method between the affinity and the trizol method for the isolation of RNA? Okay, so you understand the difference between the difference between the trizol method and affinity the method. So trizol. So if you want to isolate the messenger RNA, right, you have actually two methods. One is the trizol method, where you are going to use the triazole, and the other method is where you are actually going to use the affinity beads, right? Now, affinity beads, uh, where your affinity beads either could be magnetic beads or the normal beads, right? And affinity beads are actually going to have a bead on which you are actually going to have the uh, T nucleotides, right? So these T nucleotides are actually going to interact with the A nucleotides because all the messenger RNA will have the poly A tail, and as a result, they are actually going to bind, and then ultimately you are going to have the um, competition. So you are going to put the high salt and other kinds of conditions, and as a result, it is actually going to release the messenger RNA, which is going to be poly A tail, right? This is affinity chromatography. With the triazole method, there's no such requirement. You just are going to do the lysis, then you're going to add the triazole, and ultimately it is actually going to give you the messenger RNA pool, right? Now, this method is easy, right? Triazole method is easy compared to the performance of the affinity chromatography, because affinity chromatography requires the beads, then you require the all those kind of, suppose you are using the magnetic beads, then these are, you also require high power magnets to recover the beads from the solution and so on. And this is more time consuming. So it is a time consuming process compared to the triazole method. And that's why the affinity chromatography method is less popular compared to the triazole method. So almost 90% people are doing the triazole method compared to the affinity chromatography method. But affinity chromatography also has the advantage that it's actually going to give you the more pure messenger RNA. It's not going to give you anything else except that compared to that the triazole method, because triazole method there could be a possibility that you may actually get the contaminating or similar similar type of RNA molecules. So is it clear, Sarmita? Okay. Now we have another doubt that sir, why there are three different types of DNA polymerase in E. coli? So I'm sure you know that we have the DNA pol 1, right, and pol 2 and pol 3 in the bacterial system. So the function of these pol 1, pol 2, pol 3 are different, and their efficiency and their affinity for the polymerization is also very different. And some places you require a slow polymerization, some places you require, and it's always be a kind of a backup plan, right, in the cell, that if suppose pol 2 is not being able to synthesize because of the one or other problem, then pol 1 or pol 3 will take over the questions, right? This is what exactly happens when you are talking about the bacterial system. Bacterial system does not rely on the single machinery, right? It always have a backup. So in case you are going to do something with the pol 2, then the pol 1 and pol 3 will take up. If you do something to the pol 3, then pol 1 and pol 2 will take up, and so on. So that is could be the one of the reason that they are actually uh, that you are actually going to have the different types of DNA polymerase. Then we have another question. Kezia, good afternoon. And if you have any question, okay, and I have another question from the Ashwarya. So Ashwarya is saying, what is the role of the chylomicrons? So chylomicron are chylomicrons are actually the carriers, right? They are actually, their main job is to carry the lipids, okay, from uh, from the one part of the blood. So they are actually the carriers. So they will actually going to deliver the lipids to the organs. Okay, and at the same time they are also going to take up the lipids from the organ and put it into one of the major organ is the liver itself. Okay, so chylomicrons are going to be take up the lipids from the liver, then they will give it to the other organs, and from the other organs they are also going to carry the lipids and they will give it to the liver, and they are actually be the major molecules which are responsible for the fatty livers. So if there will be more number of chylomicrons and you are actually going to have the more inflow towards the liver, then it is actually be responsible for the fatty livers. Then we have another question from the Gayatri that she wants to know about is the downstream molecules I was discussing in the lecture, right? So I think better you should first understand or go through that lecture and then tell me what you want to discuss with me, because this live session is very limited. So I cannot discuss the all the molecules, downstream molecules, and so on in a limited time frame, because we only have half an hour or 45 minutes for discussion. So these kind of big topics we cannot take up into the this particular type of live sessions. But the same is true for the Harshita also. So she's saying that you explain the biosynthesis of histidine. So I think there's nothing what you, if you are not been able to understand something within the biosynthesis of histidine, then probably we can discuss that. But discussing the whole process and other things, it is beyond the scope of the live sessions. Live sessions, the purpose of live session is that we should actually discuss something which you could not be able to understand within the topic. Okay, it's not like I'm going to elaborate the whole topic with because that anyway I have done it. So if you go through the lecture you could not understand, okay sir, this step I could not understand or that step I could not understand, then that we can discuss, and we can discuss in detail, not the whole process. You cannot say, sir, I cannot understand glycolysis, so please explain the glycolysis. That is not what the purpose of this live session actually. And I think on the live on the team actually, some of the students, I think in the beginning they were asking about this, what is the scope of the MS molecular biology, MSc and all that. So this course, molecular biology, the course is very good actually, because it covers almost more than 50% content for your GATE exams. Especially, so if you go through the content, and then if you are actually going to practice the question papers or questions from the GATE exams or previous GATE exam papers, then I think you will be able to cover almost 50% content. Apart from this, if and that may help you in terms of clearing the GATE exams. Now, second question is whether there will be a scope of doing the MSc in molecular biology or related topics. So, MSc is just the basic degree actually. You are supposed to do PhD and you're supposed to be an expert in that particular area. Then only you people will actually going to give you the job in that particular area. Simply by doing the molecular biology will not get you the the good job in this particular field. So I hope this is clear. Now we'll Shalini, I can explain in Hindi also. I am very good in Hindi also. So don't be. I don't have any problem. You ask me the questions. I'll try to address the things in Hindi also. Shalini, do you have any question? Okay, so Ankita is asking what is the role of the carnitine acyl transferase 1 and 2. So carnitine acyl 1 and 2 are actually the transporters, right? And they are see lipid molecules, right? Especially the fatty acids like acetyl CoA and those kind of molecules. When they are being produced, so lipid molecules are going to be get into. So when you want to generate the energy from the lipid molecules, uh, you are supposed to put into the beta oxidation, right? And beta oxidation, all the enzymes of beta oxidations are present inside the mitochondria, right? So, and the lipids is actually being stored into, or it is present in the cytosol, right? So, from the cytosol, the first job is that it has to be enter into the mitochondria. Then only there will be a beta oxidation, and then only you can be able to generate the energy, right? Energy in terms of the ATP, right? So how you are actually going to transport the lipid across the plasma mitochondrial membrane, because the lipid is so that is the place where the carnitine shuttle is actually going to participate. So in the carnitine shuttle, you have the carnitine 1 and 2, and their job is to transport the lipid inside the mitochondria so that the mitochondrial enzyme like the beta oxidation will take place, and that's how the mitochondria is going to produce the energy. Is it clear? Okay, Mansi is asking, sir, can we get an internship in IITs? If we don't have any project done in past and SPI of 8.5, it would be a great help if you tell a bit. Okay, so NPTEL actually gives the opportunity for you to do the internship into the IIT, especially suppose you are doing this course and you are very good in terms of the performance. So then you can get a chance of applying for internship into my laboratory. And then I normally take students, like currently also I have 2 students who are working in my laboratory as an intern, which are from the NPTEL. And the best part is that when you do the internship through through the NPTEL program, you will get the stipend also. And but we don't consider your prior CPI or SPI. So it hardly matters whether you are a 10 pointer or 9 pointers or 7 pointers. If you are doing good in the course, then probably we will consider you for an internship, not because of your previous performance. So in the course, you should be performing good rather than the previously. Do we have any more questions? Okay, so Harshita is asking how we can apply for the internship. So once the course result will be out, then for example, this course is running in the summer, right? So this course is running in the winter. So the result will be out in I think April, right? So once the results are out, then I think they will actually going to have a call for internship, and then you can actually be able to apply for an internship. And then once you apply for an internship, all those applications will come to me for me to select. And I normally select based on your performance in the course, whether you are a topper or the gold certificate holder or platinum gold certificate holder and so on. And sometimes it depends on the students prior discussion with me also, but mostly it is based on your performance. So once the results are out, then you can apply for an internship. And those who apply for an internship, their application will come to me according to how many slots I will provide to the NPTEL. and then you can be able to get into my program. So for example, this course is running in the winter, so you will be eligible for either the coming summer for a internship or the following winter. But so that depends on the NPTEL thing, right? So you can write to the NPTEL IIT Madras office, and they will be able to give you the more information about the internship program. Shalini is asking, can you explain me DNA sequencing? Right, I think that is what you're asking, right? Again, I have the same problem actually. DNA sequencing is a very big topic. Okay, but let me tell you the basic thing, right? So suppose you want to do a DNA sequencing, right? So DNA is actually DNA is a sequence of nucleotides. So for example, ATGC, ATCC, GGG, like that. Okay. So since these are repeating units, you can be able to sequence this information by two methods. One is called as Sanger's method, right? And another one is called as Maxim-Gilbert method. Now, what is Sanger's method? Sanger method is that instead of taking all the nucleotides, you take one of the nucleotide, which is called as the DDNTPs, right? So you take the DDNTPs. So, and then you are actually going to make the four reactions. Reaction for A, for reaction for G, a reaction for T, and reaction for C. So you are going to split this into four reactions. Reaction for A. So in the reaction for A, you are going to take the DDATP. In the reaction for G, you are going to take the DDGTP. Then reaction for C, you are going to take the DDCTP. And reaction for and then for T, you're going to take this. Okay. And what will happen is wherever in whatever the reaction you have taken, right, it is actually going to stop the synthesis at A. So for example, in this sequence, if you say this sequence and if you do this, then what will happen is that it is actually going to start the polymerase. So taking all these reactions and you do the PCR. And then once you do the PCR, it is actually going to stop here. If you're talking about this reaction, so it is actually going to give you these fragments. It will give you A, then it will give you TGCA. Okay, so TGCA. Okay, so these are the two fragments you will get for A. For G, it is actually going to give you this. So it will give you ATG, then it will give you CATCCC and G, right? So it will give you this fragment. So it is actually going to give you ATG, right? So whatever the nucleotide you're going to take as DDTTP, it is actually going to break. So it's actually going to break here. It is going to break here for A, right? For G, it is going to break here, and it is going to break here, right? For C, it is actually going to break wherever you have C, right? Here, here, and here. And since you only had one T, so it is actually going to break here. And then these fragments, if you analyze onto the sequencing gel, like the polyacrylamide sequencing gel, it is actually going to give you the sequence, right? And that interpretation we have discussed in detail when we were talking about. Similarly, in the Maxim-Gilbert method, instead of using an enzyme and the DNTPs, it is actually uses the chemicals. So it is uses the chemicals which are targeting either the A or T or G or C, and accordingly it is doing the same thing. It is doing the same thing and is generating the fragments, and then these fragments are also going to be analyzed, and that's how you are going to get the sequencing of these particular big sequence. Because ultimately what you're going to get is you're going to get these fragments, and these fragments then you can be able to put them into together, and then they are actually going to be you can be able to do the sequencing, right? For example, this is the sequence, then this is the sequence is common between this and this, so it is actually going to be common. And then this is a sequence between common between this and this, and so on. So this kind of sequence analysis is going to tell you the complete sequence of this given DNA. Okay. Then we have another question from the Kezia. She's saying that, can you explain the role of cytochrome C? So let me give you the. Okay, let me share this. Okay, so okay, let me share my screen again. I'm sure you can be able to see my slide, right? This is the same slide what I have used for teaching you, right? So cytochrome C is going to be released from the, so right? So cytochrome C is going to be released from mitochondria, right? And then it is actually going to form the apoptosome complex, which means cytochrome C is going to form a complex with APA1, and that is going to be called as apoptosome. This apoptosome is actually going to recruit the procaspase 9. So procaspase 9 is the initiator caspase, and it is actually going to act on the procaspase 9. So you know that the caspases are actually having the two chain, right? So it's actually going to have the inactive form. So these procaspases are present in the inactive form, and once these cytochrome C is released, it is actually going to activate the procaspase into a activated caspase, and these activated caspase are then actually going to act onto the procaspase 3, and this procaspase 3 is called as the executory caspase, because that is the caspase which is ultimately going to form, going to degrade the proteins and other kinds of biomolecules. Because ultimate goal of this apoptosis is that it is actually going to initiate the reaction so that the cellular machinery is going to be degraded, right? So that that components of the cellular machinery can be used for energy production, and they can be used for other kinds of purposes from the rest of the cells. For example, if you have 100 cells and only 10 cells are going through apoptosis, then rest 90 cells are going to take up all those materials so that they can be able to use that material. For using that material, they are supposed to degrade that material. So what they are going to do is they are going to release the cytochrome C, and cytochrome C, once the cytochrome C is going to be released, it is going to make a complex with APA1, and that is going to activate first the caspase 9, and then the caspase 9 is further going to activate the caspase 3, and then caspase 3 is actually going to enter into the DNA and enter into the nucleus, and then it is actually going to activate the caspase activated DNAs, and that is actually going to destroy the DNA. And the caspases are proteases, so they are also going to degrade the proteins, and ultimately you are going to have the complete degradation of the proteinaceous substances. You are going to have the complete degradation of the DNA, so that all the DNA is going to be formed the small fragments, and these fragments, and then ultimately these fragments are going to give you the nucleotides, and these nucleotides can be used for remaining cells. Is it clear? Okay, let's now go to the team. So Harshita, do you have any question or anything you want to ask me? Harshita, I think you are online also. So it's better to. Okay, so she doesn't want to ask me anything. Hiral, you want to ask me anything? Okay, fine, good. At least you both can unmute yourself and at least introduce yourselves, what you are doing and why you have taken this course and so on.
Good afternoon, sir. So my name is Harshita, and I'm currently pursuing BSc in microbiology, and I'm in second year.
Okay, okay. So how you found the course? How much you found useful this course for you?
So this course is very useful as I'm preparing for the competitive exams for further studies. So molecular biology holds a great.
Okay, good. I hope that you will like the course and you will register for the exam also. And Hiral,
Hello, good afternoon, sir. Am I audible?
yes, you're audible.
Um, I'm currently working as a clinical data manager at Velocis Clinical Research. For the part-time PhD purpose, I take this course.
Okay, good. So if you have any queries, you don't have to wait till live sessions. You can drop me a message in the discussion board itself, and I'm sure you might have noticed I'm pretty active in that. So and that is the place where TAs are not the monitoring that. So I'll be happy to answer all the questions in case you have any queries. In fact, you can drop me mail also if you want.
Oh, sure. Thank you.
Okay, so I think we don't have any more questions from the student side. So I think it's better to wind up now. So thank you for your interest, and I hope you will be keep learning the new things in this particular course. I have tried that we should give you not only the theoretical knowledge but also the experimental knowledge. So in due course, you will see when the we'll move further up. You will see that how you're going to see a lot of experiments in the course also. And if you have any queries, you don't have to wait till the live session is coming and all that. You just drop me a message or drop it into the live discussion board, and I'll be happy to answer. So with this, I would like to conclude today's live sessions. I hope you will enjoy the course and keep learning.